My name is Catherine Losinski, and the date is 10-8-24. The first case that I chose to unravel was case number 1, in which a newborn is struggling with a potential urea cycle disorder affecting her ammonia rates, which are toxic, especially to the brain, especially at that age. So the first question asks to discuss the normal rates of ammonia transfer in the body that should be occurring, including certain enzymes, and to discuss the role of transamination. So, glutamate is an amino acid that comes from the muscle and can be deaminated by an enzyme called glutamate dehydrogenase, which I have represented as this picture here. This is also a reversible reaction that occurs in the liver that causes glutamate alongside NADP to convert to alpha-ketoglutarate, NADPH, and free ammonia, or alpha-ketoglutarate, NADPH, and fixed ammonia to convert to glutamate and NADP. Since it is a reversible reaction, this reaction can result in the freeing or fixing of ammonia. If the ammonia is freed, as shown in this reaction, the ammonia then enters the mitochondria in the liver and enters the urea cycle to be detoxified and converted into urea. The urea, which is less toxic compared to ammonia, then enters the blood, is filtered in the kidneys, and then excreted as urine. Glutamase is another enzyme that frees ammonia. This enzyme is mainly located in the liver and the kidneys and breaks down glutamine into glutamate and also ammonia. In the liver, the ammonia then enters the urea cycle to be detoxified, and while in the kidneys, the ammonia excreted into the urine, as shown here. In contrast to glutaminase, glutamine synthase, as shown here in the brain, fixes ammonia through the conversion of glutamate to glutamine. This enzyme is active in peripheral tissues, as shown here in the brain, to aid in storing the toxic ammonia in glutamine. This glutamine can then travel into circulation to the liver, where it can be acted upon by glutamase, and the free enzyme can enter the urea cycle. Transamination is a process in which there's the transfer of an amino group from an amino acid, such as aspartate or alanine, as shown here, to a keto acid, such as alpha-ketoglutarate. When these amino acids go through the process of transamination, there is no release of free ammonia, maintaining the toxic chemical instead of releasing it into circulation. So, for example, on this slide, alanine aminotransferase, which is shown as this red enzyme, is an enzyme that partakes in the transamination reaction that works with the cofactor PLP to transfer the amino group from alanine to alpha-ketoglutarate, forming pyruvate and also glutamate. During fasting, the generated pyruvate, as shown here, can then enter the process of gluconeogenesis and become glucose to provide energy. If the liver does not immediately need glucose, the pyruvate can become acetyl-CoA and enter the TCA cycle. But if there are enzymes found in the blood which aid in these transamination processes, such as ALT or AST, this is indicative that there is liver or heart damage in the body. Nitrogen transport is also greatly connected to gluconeogenesis in the fasted state. So during fasting, proteins break down into amino acids and then enter the process of transamination, transferring the amino acid to alpha-ketoglutarate and forming glutarate, as previously described on slide number 4. The resulting glutamate can then donate its amino group to pyruvate and result in alanine. The alanine glucose cycle then takes place, in which alanine is transported to the liver and transaminated back into pyruvate, which is used as a substrate for gluconeogenesis and produces glucose. So that is shown right here. During transamination reactions, ammonia is released and can enter the urea cycle for excretion. This cycle continues, and the resulting glucose can enter the blood and then re-enter the muscle for this process to take place again. Since farther analysis of the patient shows high urine orotic acid levels and normal plasma sertraline levels, the urea cycle enzyme that is most likely deficient is ornithine transcarbamylase. This enzyme combines ornithine, which is shown right here, and carbamoyl phosphate, which is shown right here, and produces sertraline. If there is a defect with this enzyme, carbamoyl phosphate builds up in the mitochondria and enters the pyrimidine synthase pathway instead of continuing on into the urea cycle. When it enters the pyrimidine synthase pathway, orotic acid builds up, which is an intermediate for this pathway. Sertraline levels are normal because it is not a complete deficiency of OTC, which would dramatically lower sertraline levels. The urea cycle enzyme that is least likely to be impaired is arginosuccinate synthase, which is shown here on the cycle. This enzyme catalyzes the synthase of arginosuccinate from aspartate and sertraline. Since there are normal plasma sertraline levels, this points to arginosuccinate synthase least likely to be impaired in the urea cycle. Because of this, the amino acid that becomes essential in the urea cycle due to this deficiency is arginine. Typically, arginine is non-essential because it is produced in the urea cycle, but with the deficiency of ornithine transcarbamylase, the synthesis of this amino acid becomes more difficult, resulting in it becoming essential and needing one to consume it in their diet. The next case connection case I chose was case number 3. This case is about a 47-year-old man with severe pain in his right great toe, and he has had labs drawn and is diagnosed with gout. Gout is a condition in which the level of uric acid in the blood is high, characterized by hyperuricemia. Uric acid is a waste product of purine breakdown, and the body normally can eliminate it through urine. But if there's a surplus of purine breakdown or the kidneys cannot filter it properly, the uric acid can build up in the blood, causing a greater rate of urate levels. If too much uric acid is in the body, crystals can form, which can cause inflammation in certain body parts, seen in this patient's great right toe. In labs, gout is characterized by plasma urate levels greater than 7.0 milligrams per 100 milliliters, in which the patient has a greater rate of 10.5 milligrams per 100 milliliters. Gout is also characterized by 413 plus or minus 78 milligrams per 24 hours of uric acid found in the urine. This patient is underproducing uric acid in the urine at a level of 215 milligrams per 24 hours. The best explanation for the cause of gout in this individual is decreased urinary excretion of uric acid. The low excretion of uric acid suggests that the individual's kidneys are not functioning as they should and cannot excrete the urine acid out as urine. This could be because of a disorder in the proximal renal tubule, which, shown here, is the site uric acid reabsorption. If there is a dysfunction in the proximal renal tubule, such as hyperactivity, there could be excessive absorption of uric acid, elevating plasma urate levels and decreasing uric acid levels in the urine. The overproduction of purines is most likely not the cause of gout in this patient, because if purines were being overproduced, the labs would show both an elevated level of plasma urate and urinary uric acid excretion. This is due to when purines are broken down and enter through a series of enzymatic reactions, but they ultimately produce uric acid in the blood. While this is consistent with the results of the plasma urate levels in the patient, what is not consistent is low levels of urinary uric excretion. If there were a surplus of purines being broken down, there would be a surplus of uric acid found in the urine as well, which is not true in this patient. Decreased salvage of purines is also most likely not the cause of gout in this patient. The salvage of purines is a process of recycling existing nucleobases from the diet or nucleotide catabolism to produce nucleotides in an energy-efficient tumor manner. If this process decreased due to enzyme deficiency, the plasma urate and urinary uric acid levels would be increased in the patient. This is because in the salvage pathway, these purines are reused and not broken down into the uric acid. So if there is a decreased salvage of purines, more purines are broken down, resulting in an increase in uric acid in both the blood and the urine. For example, in Lesch-Nyhan syndrome, the enzyme HGPRT is deficient, which saves purines and reintroduces them into the purine synthesis pathway. Patients who have this syndrome show increased plasma urate and urinary uric acid levels, and this patient only shows increased plasma urate levels. The potential impact of high intake of fructose via the diet can increase or worsen the initial condition of gout in this patient. The patient stated that he switched to regular soda because the fake soda was quote-unquote worse for him. Regular Coca-Cola contains 54.2 grams of fructose per liter, while Diet Coca-Cola contains no fructose, but artificial sweeteners instead. So, while fructose is metabolized in the liver and converted into fructose 1-phosphate by fructose kinase, large amounts of ATP are consumed, and the result is ADP. ADP is converted into AMP, and as ATP levels decrease, AMP goes through the steps to be converted to uric acid. This is why fructose metabolism can ultimately increase uric acid production and thus lead to the formation of crystals in the body, leading to gout. This could especially have made an impact on this patient because he drinks between 4 to 5 liters of soda a day, which is an excessive amount of fructose for the body. A high intake of fructose can also affect the kidneys. Fructose leads to the increase of uric acid reabsorption in the proximal renal tubules, which we referred to earlier. This reabsorption elevates the reabsorption of uric acid in the blood, leading to a decrease of uric acid in the excretion of urine. Since this patient has had high plasma uric acid levels and low urinary uric acid rates, his excess of soda drinking could have highly led to the worsening of his condition.